I'm pleased to present our CVPR 2025 highlighted paper, Do Computer Vision Foundation Models Learn the Low-Level Characteristic of the Human Visual System? The human visual system has been exposed to natural images since birth, gradually developing key properties such as increased sensitivity in bright environments and a bandpass characteristic in spatial frequency sensitivity. Similarly, vision foundation models trained on large-scale natural image datasets have demonstrated strong generalization across different tasks. However, most existing research focuses on their task-specific accuracy. In this study, we take a different perspective to investigate these models. So this work aims to answer this question. Given that both vision foundation models and the human visual system are exposed to vast amounts of real-world images, do they exhibit similar low-level vision characteristics? So whether artificial neural networks process information in the same way as humans remains a topic of debate. Most studies suggest that neural networks exhibit distinct error patterns, for example, relying more on texture than shape in image classification tasks. However, some studies suggest that the gap between neural networks and the human vision is narrowing. Overall, prior research have primarily focuses on the consistency in high-level vision tasks. In contrast, our study is the first to examine their differences from a perception science perspective. We aim to evaluate how vision foundation models respond to stimulus commonly used in human vision research and compare these responses to human psychophysical data and tested 45 leading vision foundation models and generative models. Inspired by human perception experiments, we input the test and reference images under various conditions into the image encoders to extract feature vectors and compute the similarity score (SAC). Ideally, the countermaps derived from these scores should align with human experimental results, and to quantify this alignment, we introduce the model alignment score. Next, I will introduce each test type, the corresponding human and machine vision characteristics, and how they relate. We begin with near-threshold contrast detection, which refers to the ability to distinguish luminance or color differences between a stimulus and its background. This is typically quantified as a minimum detectable contrast, known as the contrast threshold, with sensitivity defined as its reciprocal. We propose 6 subsets covering various dimensions, including spatial frequency, luminance, and stimulus area. Examples from each category are shown in the figure on the right. This figure presents representative experimental results, with each row corresponding to a test type and each column to a foundation model. In each subplot, the x-axis denotes spatial frequency and the y-axis represents sensitivity. The black dashed line indicates human responses, while the blue-purple counterlines represent similarity scores (SAC), with purple indicating the lowest SAC. If these models exhibit human-like contrast detection, the lowest SAC counters would align with the black dashed line. From the data, we can conclude that most models exhibit bandpass characteristics, especially for achromatic stimulus. However, these characteristics are often irregular, with peak sensitivities differing from humans. Notably, for chromatic stimulus, models fail to replicate the human-like shift towards lower frequencies. Contrast masking is another key characteristic of human vision, where the presence of a super-threshold background stimulus mask reduces the visibility of the target stimulus test. This phenomenon occurs when the mask interferes with the detection or discrimination of the test stimulus, typically elevating the contrast detection threshold of the foreground. It includes 2 subsets, phase-coherent masking with a sinusoid grating background and phase-incoherent masking with a band-limited noise background. The prediction from DynoV2 and OpenClip contrast masking experiments was surprisingly consistent with the human data. Their responses roughly matched the slopes of the human data. The match was even stronger for phase-incoherent masking. Overall, the computational models performed closer to human data on contrast masking tasks than on contrast detection tasks. One possible explanation is that the signals that trigger contrast masking are widespread in natural images, while the stimulus used in contrast detection experiments are typically faintly visible patterns on a uniform background and are relatively rare in natural images. Contrast constancy refers to the ability of the human visual system to perceive relatively stable physical contrast at different spatial frequencies and brightness levels, especially at superthreshold contrast levels. And the contrast matching results are shown in the figure here, where the colored dashed line represents human data and the solid lines are the values predicted by the visual model. We found that only DynoV2 and OpenClip roughly follow the contrast constancy trend represented by the dashed lines, while the performance of other models lacks stability at different spatial frequencies. Here we show the alignment scores of all models on all tests, and there are three main observations. First, the human vision model color visibility is of course the best in most tests. And second, some variants of OpenClip and DynoV2 surpass color visibility in the contrast masking tests. And third, in fact, there are also large differences between different variants of the same model, and it's not necessarily the most complex model that is most similar to humans. We also conducted additional experiments to identify key factors influencing the similarity between models and human vision. While no definite conclusion were reached, we observed that, first, model performance on their original tasks positively correlates with their model alignment scores on the contrast masking and matching tasks, and second, model parameter numbers and computational complexity show little to no correlation with model alignment scores. And in summary, we have developed an experimental protocol to evaluate the similarity between machine vision models and the human visual system. We found that selected computational models, for example, variants of DynoV2 and OpenClip, show surprisingly high alignment with superthreshold human contrast masking and contrast matching data, but little alignment with the near-threshold contrast detection. It means that computational models don't have the same bottlenecks as human vision, but through training, they still attain invariance and efficient contrast coding that resembles that of the human visual system. Thanks to all my collaborators and thanks for listening.